This is our second podcast in our series on the eye, and in this podcast we're going to talk about the anterior regions of the eye. So we're going to be talking about the cornea, the sclera, the iris, and the ciliary body and ciliary processes. I think I told you in the first podcast that the cornea had 3 cellular layers and 2 non-cellular layers. So I've shown the cellular layers highlighted in yellow, and this one looks a little bit funny, and you'll understand why in just a second. And then the non-cellular layers I've just labeled in the white font. So we'll talk about the corneal epithelium. That's a stratified, squamous, non-keratinized epithelium. The corneal epithelium is continuous with the conjunctival epithelium that overlies the sclera. The corneal epithelial cells desquaminate, and they turn over approximately every 7 days. So there's a fairly rapid cell cycle time there, and these cells are derived from a stem cell niche, which is in the corneoscleral limbus, and we'll talk about that in a little bit. Bowman's membrane is essentially the basement membrane of the corneal epithelium. It's a barrier to the spread of infection. It does not regenerate after injury, so scar formation in Bowman's membrane can impair vision. Now, the corneal stroma accounts for about 90% of the thickness of the cornea, and it consists of many thin lamellae, which are essentially collagen bundles that run at right angles to each other, and it contains fibroblasts between the lamella. That's why we call this a cellular layer, but it's also a layer of substantial extracellular matrix material. That's why I've highlighted it in this funny font. So the fibroblasts, the cellular, the extracellular matrix material, of course, is non-cellular. So the extracellular matrix material in the corneal stroma are essentially bundles of collagen and sulfated glycosaminoglycans. And you'll come to appreciate these collagen bundles run at right angles to each other in a very precise orientation. That's why the cornea is transparent. Descemet's membrane is essentially the basement membrane of the corneal endothelium. Descemet's membrane can be regenerated after injury. And the corneal endothelium is a single layer of epithelium. It's the metabolic center of the cornea. It regulates water content of the cornea, and it has a limited capacity to regenerate. And I point out that we call this the corneal endothelium. It's really a simple squamous epithelium. It's given the name endothelium, but this is a case where these cells don't really line blood vessels or lymphatic vessels, but the term corneal endothelium is one that is just stuck in the literature. Here we're going to look at the cornea, the sclera, and the anterior portion of the uvea, in other words, the iris and the ciliary body. The limbus is essentially the corneoscleral junction. It's just a region of transition between the cornea and the sclera. So it would be in this area on the diagram, and that transition is the stem cell niche that we talked about, where the cells of the cornea come from as they regenerate. The corneal stroma, there are, as we said, multiple lamellae in the corneal stroma, and this electron micrograph just shows portions of 3 lamellae. You can note a fibroblast here at the arrow, and these collagen fibrils that are running at right angles to each other, thus giving the cornea its transparency because of the regular arrangement of these collagen fibrils. The sclera would be essentially the posterior 5/6 of the outer layer of the eye wall, and here the uvea, the middle layer. The limbus contains the iridocorneal angle, and in this area is the outflow apparatus for aqueous humor. We'll talk about that in just a few seconds, but there are endothelial lined channels of the trabecular meshwork. These channels also are given the name spaces of Fontana, and these essentially drain or merge into a scleral venous sinus canal called the Canal of Schlemm. And then the ciliary processes that are in the posterior chamber produce aqueous humor. if you allow me, let me put in the lens here. The fluid is going to pass from the posterior chamber around the lens into the anterior chamber, where it's going to be drained into this trabecular meshwork and the canal of Schlemm. So in the diagram down here, the aqueous humor is synthesized here. It's going to pass around, I'm going off the micrograph here, but it's passing around the lens, around the iris, and it's going to drain into this trabecular meshwork into the canal of Schlemm. So again, let me highlight that on the cartoon. It's synthesized here, it's passing around the lens and draining into the canal of Schlemm. Likewise here. So on the big micrograph like that, the lens not shown here, but it would be passing around the posterior chamber into the anterior chamber and draining into these trabecular meshworks in the canal of Schlemm. Now the epithelium on the iris and ciliary process consists of two layers, and that's shown in the higher power view. There's essentially a pigmented layer and a non-pigmented layer. We're going to look at this just schematically for the eye. We've already now talked about the cornea. You can appreciate this. I want to now talk about this region. We want to talk about the iris and the ciliary body, because we want to talk about how these function not only for lens accommodation, but also to control the size of the pupil. We will talk about the retina in our next podcast. So you can notice here the lens being attached to the ciliary process with these zonular fibers. A couple of other points I want to make before I go on. Here's the ora serrata. We're going to talk about the retina in the next podcast, but the ora serrata would designate the end of the neural portion of the retina like so, and here on the anterior portions we would define this as the iridial retina. We'll talk about this more in our laboratory sessions. You can see the conjunctiva and the outer layers of the cornea and sclera are essentially continuous with the conjunctiva. The iris is the most anterior part of the uvea. It forms a contractile diaphragm in front of the lens. It's got a highly vascular connective tissue stroma. The posterior surface of the iris is covered by pigmented cells, the so-called posterior pigment epithelium. The basal lamina of these pigment cells faces the posterior chamber. The anterior pigment epithelium is essentially a myoepithelium. It lies below this pigment epithelium. The myoepithelium, the cells contain pigment. The cells also contain fairly long contractile processes on their apical boundaries that extend radially, and you'll see this in just a minute or two. These contractile properties are designated as the dilator pupillae muscle of the iris, and I put that in quotes because the dilator pupillae is not a true smooth muscle. It's a contractile myoepithelium. The heavily pigmented cells in the iris make it very difficult to discern the histology of the different layers in the iris, and you'll see how difficult that is on the histological slides. The sphincter pupillae muscle is a true smooth muscle. It's in the stroma of the iris near the pupillary margin. The sphincter pupillae is circumferentially arranged bundles of smooth muscle, and it contracts the pupil. The anterior surface of the iris is a discontinuous layer of fibroblasts plus melanocytes. There is no surface epithelium on this portion of the iris. This is unusual. No surface epithelium. The number of melanocytes relates to eye color, and essentially eye color comes from light that's reflected back from the pigment cells on the posterior surface of the iris, and eye color ranges from blue to kind of a greenish blue to gray to brown. The fewer the number of melanocytes in the pigment cells, the more blue the eye would be. The greater the number of melanocytes in the pigment cells on the posterior surface of the eye, the more brown the color of the eye would be. The iris contains the dilator pupillae muscle, and again I put that in quotes because it's not a true smooth muscle, the myoepithelial cells, and then the sphincter pupillae muscle, which is a true smooth muscle. In the histological image, this is showing the sphincter pupillae muscle. The dilator pupillae is under sympathetic control, the sphincter pupillae is under parasympathetic control, and the size of the pupil is regulated by these muscles, and light sensed by retinal ganglion cells plays a role in establishing a reflex arc to control the contraction of these muscles, and we'll talk about that a little bit later. This just shows the sphincter pupillae, and the sphincter pupillae muscle reduces the size of the pupil in bright light. The dilator pupillae muscle increases the size of the pupil in low light. In the histological image, a portion of the lens is just shown here to help you orient. So on the cartoon, the lens would be here, and so it would be talking about these surfaces in the iris. The dotted line here is delineating the posterior pigment epithelium from the anterior pigment epithelium, the so-called myoepithelium. So all of this is sphincter pupillae muscle. The next image is going to show some images of the iris that are not quite at the very tip. Here's the iris, sphincter pupillae muscle here, and then a section of the iris here showing the dilator pupillae, these bands of myoepithelial cells. These are essentially long apical processes of the epithelial cells that are bending forward, and these have contractile filaments of actin and myosin in them. Contraction of the sphincter muscles reduces the pupil in bright light, and contraction of the dilator muscle increases the size of the pupil in low light. And as we said, this is controlled by a reflex arc. Light is sensed by retinal ganglion cells, and those are going to feed back through neural networks that are going to innervate these muscle groups in the iris. We now want to consider the ciliary body, and that's a thickened anterior portion of the uvea. So we're talking about this region of the uvea, and it extends essentially from the base of the iris to the ora serrata. There are zonular fibers that arise from these ciliary processes, and they function to attach to the lens. Here would be the zonular fibers originating from these ciliary processes and attaching to the lens. The ciliary body is layered similar to the iris. The stroma has an outer smooth muscle layer, which is the ciliary muscle, and an inner vascular layer. The epithelium covering the internal layer is continuous with that of the retinal epithelium, and it's essentially the non-neural retina, and that would be in these areas that are anterior to the ora serrata. Now, the functions of the ciliary epithelium are to secrete the aqueous humor and to secrete the zonular fibers and the anchoring of the zonular fibers into the lens. The ciliary muscle is organized into functional groups, and it's difficult to discern those functional groups histologically. There are meridial groups, so-called longitudinal, and these stretch the choroid and they may help to drain aqueous humor. There's a radial or oblique group. Contraction of these muscles flatten the lens. That allows the eye to focus on distant objects. And there's a circular or a sphincteric group. Contraction of these muscles reduce tension on the lens. That allows for accommodation for the eye to focus on objects that are close by. When the radial muscles contract, the circular muscles relax. When the circular muscles contract, the radial muscles relax. Accommodation is based on the action of these muscles, and this cartoon shows accommodation based on the action of the circular muscles. Here, when the circular muscles are contracted, the zonular fibers are relaxed, the lens gets fatter, and that's necessary to have near vision. When the circular muscles are relaxed, the zonular fibers get under tension. That pulls the lens flat, and that's necessary for far vision. Now note, as we said, it's difficult to discern the histological groupings of the ciliary muscle. The ciliary muscle is shown like this, but you really can't discern histological groupings of the different ciliary muscles. And the ciliary processes here, remember, secrete the zonular fibers and the aqueous humor. The outflow pathway for the aqueous humor, it's synthesized and secreted by the ciliary process, and it's going to flow, as we've already hinted, past the lens, around the iris into the anterior chamber to drain into the trabecular meshwork and canal of Schlemm. That's just shown here. Aqueous humor synthesized and secreted here, draining past the lens. Here I'm coming off the micrograph, obviously, but into the anterior chamber to drain into the trabecular meshwork and the canal of Schlemm.